everyone. Thanks for coming to Eco Lodge. First off, we'd like to thank our sponsor, Carib Trans, for live broadcasting our event. So also welcome to all of our virtual visitors. And of course, we'd like to thank El Momo Cottages. Andres is in the audience tonight. Thank you for hosting, Ian. We'd also like to thank our two big sponsors, public entity Saba and Prince Bernhard Culture Funds. So round of applause for them. And our program would not be possible without the help of our 50-plus sponsors on the island, on St. Martin, in the States, St. Croix. So big round of applause for them as well. Raffle tickets. We are selling raffle tickets all month long, so you can stop by Lynn at the bar here. Pick up your raffle tickets, only $5 each, $10,000 worth of prizes. Final draw is October 27th. You do not have to be present to win. Our upcoming events, our next presentation is at Long Hall, 5:30 p.m., and it is with our iguana expert, Natalie, who is also in the audience. Natalie, if you can give a wave. You can also then join her for a hike to see iguanas and see the difference between the native species, invasive species, and hybrids potentially. And then our following presentation is Sunday at Queen's Hotel, which will be with our coral restoration expert, Aaron Pilnick. And if you join any of these field projects, you can get a free t-shirt or tank top. So be sure to join, and the t-shirt is yours. They're made out of recycled plastic material. And cloud top. Who has a cloud top tonight? Yes, yes. We will be selling them all month long. You can purchase them also at bars and restaurants on non-presentation nights. And then I will introduce our first speaker of the night. Laurel Allen is from the California Academy of Sciences. We have partnered with them on several things, and this first thing she will be talking about is our new app or platform that they have been working on for us for 2 years? One year. But we are ready to launch, so without further ado, welcome Laurel.
Wait, let's give it up for Emily, hype woman extraordinaire. Never gets enough applause. Okay, hi, I'm Laurel Allen. I am a full-time science communicator at the California Academy of Sciences. And tonight I'm going to take just 15 minutes to tell you about the very first iteration of this new digital platform we built and piloting it right here on Saba called IsleExplore Saba. And it was really designed to center island nature and island communities in a new way. And I'm basically just going to give you a little bit of context and then walk you through it. And I just want to point out the soft launch sign. That's because we launched this thing 2 days ago. And so what the reason we're kind of sharing it tonight is because we'd really love all of your help, like checking it out, going through it, and over the next month letting us know if you see things that need to be fixed or things that could be better. And then starting in kind of November, we're going to work with a range of on-island partners to start promoting that more publicly. So, and there's parrots around. Okay, so for a little bit of context, this is the Academy. We are based in San Francisco, California, and we're a science museum. Anyone can come and visit us. There's a rainforest, planetarium, aquarium, natural history museum, and so that's the museum itself. But we are also a research institution, so we're home to about 100 in-house scientists in 8 different departments, scientific collections totaling about 46 million, and this is us kind of at home. But of course, these days, it's not enough to stay at home anymore. You need to have an impact in the world. And to kind of attack the pieces of the environmental crisis that we thought we could make a difference on, we have 3 global initiatives that are running right now. The first is Thriving California, and that encompasses the whole state of California, which is a biodiversity hotspot and is being hit really hard by climate change. The second is Hope for Reefs, which is working in lots of sites all over the world with a range of tools and programs and really cutting-edge science. And the third is the reason that we're here tonight. This is Islands 2030, and the goal of Islands 2030 is to halt biodiversity loss in 5 tropical island archipelagos by the year 2030 in partnership with local communities. And one of the co-directors of Islands 2030 is in the audience tonight, Dr. Lauren Esposito. Some of you may have seen her speak in past Sea and Learns. And so a lot of you can probably answer the question why islands for yourself, but just some really good takeaways or key or top notes. Islands are actually home to the greatest concentration of biodiversity on that's Bella. Greatest concentration of biodiversity on this planet, which is amazing and remarkable, and also really scary when you consider that 75% of all recent bird, reptile, amphibian, and mammal extinctions have actually taken place on islands. And 85% of remaining reptile, amphibian, and mammal species are highly threatened, as well as half of all birds. And so islands are incredibly valuable. They are also places that scientists have always learned a lot and continue to learn a lot. If we can figure out how to do targeted interventions on islands that actually address biodiversity loss, we can potentially scale that to other parts of the world. So they're really, really critical in this race to understand how to protect what's left. And for that reason, the Academy has actually worked on islands for over 100 years. So this is a snapshot from our 1932 expedition to the Galapagos, but we actually started working there in 1905. I don't know if anyone here has ever been to the Galapagos, but if you have, you've probably passed through Academy Bay, which is a very old legacy of that work. And that long-term work and long-term relationships is really a hallmark of each of the 5 archipelagos in which Islands 2030 is working. So we have the Galapagos, Sao Tome and Principe, Madagascar, the Philippines, and of course the Lesser Antilles. And our scientists have worked in all of these places for decades, but they've kind of worked alone. So they've just kind of been doing their own science in the field, working with local communities. But Islands 2030 is really special because for the first time, we're pulling all of this together into one initiative and one push, and we're also putting the full strength of the Academy behind it. So that means not just scientists, but also science communicators like me, and people who do exhibits, and people who do education, and people who do marketing and promotion, and all of the things that you need to really, really move the needle. So that's what makes this initiative special. And what my team does, my team's called Digital Engagement, and what we do is essentially we're storytellers. So we're telling the stories of this work and the people involved and what's at stake across all of our digital channels, and really trying to connect the public and new audiences to what's happening and why it matters, and give them ways to care about it and take action. But from the start, we knew that doing that kind of storytelling just wouldn't be enough to actually really make a difference. So the first thing that you kind of do when you're like, okay, how do I have an impact in a space, is you take a look at what's already in that space and try to identify some gaps. And one of the first things that we saw was that in the digital landscape, there are a lot of apps out there about islands. Like almost every island has some kind of app associated with it if you look in the App Store. But those things are, so while those things are not rare, they're also actually really not great. Like they're all pretty terrible. And that's because they have little to no information about nature or biodiversity. Usually nature is just kind of this beautiful backdrop. Island communities are almost totally erased. So if you look at a lot of these apps, it's like no one actually calls those places home. They're largely unauthorized. So a lot of times they're just like third parties who copy paste the tourism website and then monetize it. They're not useful to island communities. They're just designed for one and done use by tourists. So if you're visiting, you might download it, and then as soon as you leave, you're gonna delete it. And last is they don't work across all devices because they go through app stores. They're really inaccessible. So if you're someone at home who wants to explore on a desktop, there's no way to do that. So as we thought about these limitations, we kind of started to have an idea about something that would work to really counter all of these negatives. But we needed a place to test it, and we needed a place to kind of pilot it and think about how that would work. And some of you may know this woman. Who is that? So we had worked with Lynn for several years. We'd been part of See and Learn in the past. And Lynn reached out and just was asking whether we knew anything about apps, because they have a really cool project some of you may know about called Adopt a Box, and we're looking for ways to communicate that better. And so we all got together and we talked some more, and we realized there was some really nice synergy there. And of course, Saba was kind of, a 5 square mile island packed with pristine biodiversity and really friendly communities. It was a great place to pilot a project like this, so it was a really, really good fit. And going into it, we knew that we wanted our criteria to really be counter to the things that we thought were flawed about existing apps and digital platforms. So going into it, we said our goals are to really make island nature and island communities the focus of this platform. And next, we wanted to co-create this with the community. We didn't want to come in as kind of outside partners and decide what stories to tell about a place. We wanted the people on the island to tell us what that story was. We really wanted this to be useful to all audiences. Like tourists, for sure, they're critical to island economies. That needs to be a priority. but also parents at home exploring with their kids, or educators in classrooms, or like community and cultural organizations that are really the lifeblood of a place, giving them more attention. We wanted to be sure that we were doing some kind of service to all of these groups. And we wanted it to be incredibly accessible. So we don't care what kind of machine you're on, what kind of OS you have, any of that. We want it to be something that you can sit down and use and engage with. And that meant that's when we kind of decided we're not going to create what's called a native app, which are the ones that go through app stores. We're going to create this as a mobile optimized web platform that's going to be really streamlined and easy to use. And then last, really the most important thing, it's going to be free for partners and users. This product will never be monetized ever. And so with that, excited to, and again, like, let's keep in mind this just launched, but these are screenshots. When you actually look at this online, it's kind of animated and more engaging. But this is IsleExplore Saba. And so these are just kind of standard welcome pages. You can see it translates kind of beautifully to mobile. And it may be too small for you to see, but I'm just going to call your attention to the 4 main sections. And those are flora and fauna, trails and dives, community stories, and nature and community orgs. And really the heart of this platform is the flora and fauna, the nature, and community stories. So I'm going to start with those 2, and I'm just, for the rest of this, just going to walk you very quickly through what you can see when you log on and explore. So here's our flora and fauna hubs, and we've separated into 3 groups. There's species on land, in the water, and in the air. It's not super scientific, but it is really accessible. And what you see when you look at this is they're just some of the species featured. And we will be continuing to add content to this throughout the next couple of months, so you'll see even more than what's there now. But you see all of these beautiful species. And again, looks really good on mobile. And let me just walk you through a page real quick. Obviously beautiful imagery. This is Kai Wolf from the Saba Conservation Foundation. And all of the images in this app are actually either, for the most part, either Kai, or they are iNaturalist images. Does anyone here use iNaturalist? Okay, fantastic. So you'll see a lot of that here, and you'll see integration as well that makes it really special. Okay, so beautiful photos. Obviously, we're going to talk about it. And then down here, we have these modules for native and endemic. And these are things that you don't see talked about in platforms like this a lot, but they are part of what makes islands so, so special. So we really want to call attention to those 2 groups of species, and we explain why that matters. More beautiful. This is just an excuse to show off this bird, because, like, oh my God, look at it. And then we have iNaturalist observations. So this is a module that shows observations of this species actually made here on Saba. And then the little circles may be hard for you to see, but this map is actually a visualization of everywhere that this species has been seen on the island. And when you're going through all of these pages and you're exploring this, you're really getting a completely different sense of just how rich the biodiversity here on Saba is. And that's pretty exciting. And then this other module is called Ask a Scientist. So for every species page that you see, there will be a module at the bottom where a scientist is talking about that species or talking about that group of species in a really personal and accessible way. And right now, most of these are populated by our curators, like this is our curator of ornithology. But we do intend to start swapping that out so that we're really highlighting and centering scientists who are directly connected to Saba or to the Lesser Antilles. All right, let's take a look at community stories. So the people that you see here are just some of the people who gave my team their time for first-person interviews when we were here a few months ago. And if you go through this section and you watch some of these videos and you get to know some of these people, and if you live on Saba, you know some of these people already. But I really think it gives you a completely different sense of people and place when you actually hear that person telling their own story. And it's really powerful. And again, it translates really well to mobile. I'm going to take a look at one of them here. This is Elka. I don't know how many folks know her, but she's incredible, and she holds so much Saban history. What's that? Oh, yay. Okay, Henrietta's on here too. So she gave us her time, and she did this beautiful interview, and you can watch that there. It's also for people who would prefer to read. That's also written out. And for every one of the community stories, we also have a module called Ask a Local. And so these are taken from their interviews, but they're questions that anyone might have about Saba, and then they're giving them their own personal answers. So again, go in there, see who you know, see who you've met, meet some new people, and I really think at the end of that you will have a different kind of connection to place. You won't just learn more about Saba, but you feel connected in a different way, and that's just kind of how humans work, right? We tell each other stories, and then that carries meaning. And these Ask a Local modules that are scattered throughout the site, they're kind of another piece of the heart of this. Like, it's just a really different way to connect to place, and you'll learn some stuff. Some stuff is just funny. Some are stories about people growing up, or building things that you use every day. And yeah, it's great. I'm really eager to hear what people think about that. So moving into the hikes and trails, this is kind of an example of a trails page, and it's pretty straightforward, but beautiful imagery, a description of what you'll see as you experience that trail, beautiful gallery, and then information just about difficulty and distance and resources from the Saba Conservation Foundation. And the dive pages, oh no, sorry, this is a shout out to Adopt-A-Box, which was, a big, an important piece of how we got started. And this is one of the actual walks that you can do in the hikes and walks section. And again, translates really beautifully. And there are some trails on here. This is one of the sections that we're still building out. And that's also true for the dive section. Looks pretty similar, except instead of the static gallery, you see observations of species that have actually been seen at these dive sites, as well as a different set of resources. And then the last section is nature and community organizations. This is the Harry L. Johnson Museum as an example. So here again, the images, this kind of text, the descriptive stuff, but we also center a video of someone who works or volunteers there, which again gives you a different connection to place. And then these people are in the Ask an Expert module, so they're talking about their own organizations in a way that, these kind of organizations really, they are the lifeblood of a community in so many ways, and they get so little attention, so this is a really nice way to learn about that. And most of these are places you can actually walk into. So, the Saba Conservation Foundation, you might not think to go visit their office as a tourist, but it's a great resource and a great place to stop in. So you get all of that as well. And I promised 15 minutes. That was 15 minutes. We're eager to hear about the urchins. So this is a thank you to all of these organizations who have been so helpful as we did this work, both in advising us and in facilitating opportunities and just making stuff possible. So See and Learn, of course, Saba Conservation Foundation, the Saba Tourism Bureau, Queen Wilhelmina Library in the bottom, and these iNaturalist users who did a ton of heavy lifting in terms of photographing stuff here in Saba. The first one, Reef Guard, is Kai Wolf again, so he's absolutely everywhere. But we couldn't have done it without the support of these folks. And this is just a quick thank you to, these are all the IsleExplore people at the Academy. And that's really it. So again, the real point of this presentation is to let you know it's there. If you have your phones, maybe you want to take a picture of this slide just so that you have access to the web address later, or you can try the QR code depending where you're sitting. But what we'd love for you to do is just go in, have a look around, see what you think could be better, see what you think is missing. If you see something spelled wrong, please let me know. And at the bottom of every page in the footer, you'll see an email address where you can send that feedback, and that goes directly to my team. And we're really grateful for your help and support, and would love to hear from you. That's it. Thank you.
Can we please give another round of applause to California Academy of Sciences, Laurel and her team. We are super thankful for this wonderful platform for Saba. It's a great new marketing tour for tourists and for locals as well. Now for tonight's keynote speaker, Ian Hewson. Did you know that our speaker tonight doubles as a detective? He also happens to be a colleague of one of our very own resident scientists, Alwyn Hickelma, who unfortunately is off island. Otherwise, he would be here tonight. Ian Hewson is a marine ecologist in the College of Agriculture and Life Sciences at Cornell University. His research centers on the role of microorganisms in mass mortality disease, discovery of novel pathogens of marine invertebrates, and surveillance of marine viruses through metagenomic approaches. So can you guess what tonight's speaker, what mystery he solved based on all that information? You got it, Tim. All right, so give a big round of applause for Ian.
Oh my goodness, thank you for that introduction. Whoop, let me just move this up a little bit, otherwise we won't tune in. Thank you for the introduction. Thank you everybody for coming, and thank you to my hosts at El Momo and sponsors. It's been a wonderful time since I've been here, and I wish I could stay longer, but unfortunately I'm in the middle of a teaching semester where I have about 60 undergraduate students that are taking classes with me at Cornell at the moment that I have to get back to. So tonight's presentation is obviously about the long spine urchin mass mortality of 2022, which I think you probably have heard a fair bit about just in the last year. This is a big disease which has occurred throughout the entire region, spanning everywhere from Florida down to the ABC Islands and over to the Mexican Caribbean. And what I'm going to do tonight is to talk about how we actually came up with the final result, which is what's causing it, and also to give you some indication of where we're going with it and what we can do in the future about it. But I always like to start off talks with, why are we interested? All right, why do we care about this group of organisms known as echinoderms? All right, echinoderms simply means spiny-skinned organisms. It includes things like starfish and sea urchins and sea cucumbers and brittle stars. In the environment, they play extremely important roles. Basically, these organisms control the balance of different types of organisms through their herbivory as well as through their carnivory. So, for example, sea stars play a really super important role in grazing out bivalves and other types of tiny little snails and other things, and therefore allow big kelp forests to persist on the west coast of North America. In the coral reef ecosystem, you have these diadema, which basically graze out algae and allow corals to persist because they compete for space. It's worth noting that echinoderms are also economically important, all right? Not just for tourism value in a place like this, but also as a direct consumption. People actually consume things like urchins. They also consume things like sea cucumbers. And if you put a total amount on the harvest every year of, for example, sea cucumbers in East Asia, we're talking at about 21,000 tons of it a year, which is being consumed. So quite a large industry. Echinoderms are also super interesting animals because they have a very unique anatomy. Whereas most other animals have sort of a bloodstream going through them, they have, we have blood flowing through our veins. Echinoderms don't. They actually have seawater flowing through their bodies, okay? And that makes them really, really unique in terms of the types of habitats in which microorganisms, their microbiome, can actually exist. And as a result, we actually find that they have very unique microbiomes, okay? Most people are familiar that we have a bunch of bacteria and other microorganisms on our tissues and within our guts and in our respiratory tract. They're completely different in echinoderms compared to most other marine animals, and it probably is because of this very unique anatomy. But probably the biggest reasons that we are interested in them is that they experience mass mortalities. They undergo this boom-bust cycle. They become super abundant in a location, and then they just suddenly all die off, and that's been known from quite a few different systems. Before coming to the diadema disease in the Caribbean, I've actually worked on sea star wasting on the west coast of North America, a hugely deleterious event which actually started to occur in 2013. And we still don't know exactly what caused it, but the point is that wiping out 98% of the starfish on the west coast has led to some pretty huge changes out there. And so when the opportunity arose to become involved with this research in this region, I totally jumped on it. It also means I don't have to wear a thick wetsuit every time I go in the water. All right, so just to sort of emphasize what diadema actually does, here's a typical place where we find diadema. This is actually in Aruba, okay? So we have a coral head here. This is a type of coral that we call Siderastria, all right? It's the lesser starlet coral. Diadema, this is taken during the day. It's actually hiding underneath that coral boulder. And then because it's a little bit green on this projector, you can't really see it too well, but all of that stuff, that black stuff around there is macroalgae, is turf algae. And so what diadema does is it actually grazes upon that algae, and that algae would otherwise smother that coral, all right? So they play this super important role in maintaining coral reefs. And when they disappear, the reefs tend to decline. So basically, diadema keeps coral reefs healthy. Now, the first time that diadema died off was absolutely catastrophic. It was back in the early 1980s, the summer of 1983 to 1984. It was an event that was first noted by a friend and colleague of mine, Harris Lesios, in the Smithsonian Institute in Panama. And as you can see, those numbers on this map correspond to where it occurred in sequence, all right? So it started on the Caribbean coast of Panama. It then made its way to Jamaica, all over the Mexican Caribbean, up into these islands as well. It's sort of broadly following water current patterns. The key thing about it is it wiped out, 99.9% of all the diadema across this entire region. There were none here for a very long time. In the 30 years since this event, they've recovered maybe to about 5 to 10% of what they were before the 1980s. And so as a result, many of the reefs around the Caribbean really suffered. The corals do not look the same now as what they did before the 1980s. They're all completely changed in in their composition, and they're also very unhealthy. All right, so diadema, this big loss, really had a huge impact. We still don't know what caused this. All right, 30 years of research, we still have no idea what actually caused this, other than potentially it was something moving in these water current patterns. And unfortunately, a lot of specimens that were collected in the early 1980s were lost along the way because of hurricanes that moved through and destroyed a lot of institutions where they were being housed. So we can't simply go back and, probe specimens with new techniques that we have today. In April of last year, we actually had the first report of a very large die-off. It actually occurred on the St. Thomas, and then it spread very rapidly to the neighboring islands around the region. And then between then and sort of June, July, it actually had spread as far south as the ABC Islands, as far north as the Florida Keys. And there was a weird die-off that happened in the Mexican Caribbean as well, but we still don't understand how that happened or whether it was related to what was going on with the rest of the region. Looking back at citizen science records, dive logs essentially, we can say that it actually started a lot before that. It actually started in January or February, so some 3 or 4 months beforehand. So that just speaks to the importance that divers actually have in reporting these types of diseases, which otherwise we would never know about because we can't be in the water all of the time. We were alerted to this. We worked with a big consortium known as the Atlantic and Gulf Rapid Reef Assessment, or AGRA for short. They compiled maps like this where you could go and report where this disease was actually happening. And essentially we had a broad picture as to its spread around the region. From AGRA, we actually formed a big consortium of many, many different organizations, including here on Saba, the Nature Conservancy, the University of South Florida, many different academic institutions, conservation foundations. And this whole project wouldn't have been possible without collaborators getting in place, providing samples, working together, and making this research actually happen. And looking more broadly, this encompasses about 11 different jurisdictions. So Saba's down there, you can see, but we had researchers across this entire region in all of these different countries who are actively engaged in research. So these were not simply collecting specimens and sending to them. We actually engaged them in analysis of the DNA sequences that we pulled out as well. And it led to a very strong collaboration that exists to this day. All right, so bad, sort of really unfortunate thing with diadema has led to a very big, good thing in the sense that we all now communicate really well. And we have these networks through which we can traffic all of our scientific investigations. So this is what diadema looks like when it's abnormal. Essentially on the right-hand side, you have a diadema that is clinging to a rock. It's moving around on its tube feet. It uses its spine for defense. There's a couple of fish at the top there that are looking for a free meal. And so what diadema does is it typically will move its spines. They will orient them towards light and dark, and they'll fend off predators. Okay, that's the main way by which these urchins prevent being eaten. On the left-hand side, though, you can see that urchin is not doing very well. It looks really sick, right? It's actually fallen over because all of those little feet that it has, these tiny little tubes that come down from the body, they're all missing. And basically, it's just fallen over on its side. Once that happens, the whole animal is subject to anything and everything that will come along and eat them. And so when you go to an affected site, in this case, this was in St. John, essentially you'd have hundreds, thousands of these things fallen over, decaying, and we really didn't know what was causing it at the time. And in fact, the timeline of the condition going from a completely healthy to a pile of spines on the seafloor was very, very rapid. It was anywhere from a few hours from the time we first noticed it to maybe a day or two. All right, really, really dramatic condition that occurred. And like I said, once they were in the state where the spines stopped moving, where they were falling over, every single fish that's out there would basically start to pick off more spines and lead to its destruction, if you will. At most sites that were affected, it was about 90% of them. All right, so that's almost all of them. There are some sites out there where we never observed it. For example, the Turks and Caicos Islands. Never saw this disease at all. However, just in the last day, unfortunately, it's finally made it there. Okay? But point is that when it hits, it's really, really, really bad. So when they sort of formed this initial sort of collaboration to try and figure out what was going on, we had a few questions that we wanted to answer. So the first sort of order of question that everybody wants to know, scientists and citizen scientists alike, is what causes it? All right, what's causing it and what can we do about it? And so when, as a scientist, my first sort of focus goes into, is this an environmental stress? Is there something about the environment that has changed that has led to these animals suddenly dying? All right, it doesn't take much for a lot of invertebrates to make abnormal conditions that will lead to death of animals, okay? Hypoxia is a good example of that. When there's low oxygen, they simply can't breathe and they just die. Pathogens, all right? We were assuming going into this that this was some kind of pathogenic disease. It was caused by a microbe, but we didn't know which one, all right? And I'll tell you how we actually came about to investigate and find out which pathogen it was. And then we come up with some second order questions, which maybe are not a priority. So why now? Has something changed recently? Is this an invasive pathogen? If it turns out to, in fact, be an infectious agent. And then also, what can we do about it? All right, maybe that's a little bit of a higher priority, but you first have to know what's actually causing it in order to be able to come up with a strategy to treat it, okay? So we focused upon this first bit early in the investigation. We engaged with folks down here in St. John as well as in Saba, all right? So I believe that this is actually taken from Saba. And it was a big sort of effort. People were very generous with their time. Collected specimens of healthy and diseased specimens from the same site. So you went to a site, you saw some animals that were falling apart and some which were healthy, collected those, and also from reference sites, and that's an important thing. So that's a site where there's no disease at all, because we need to be able to compare what's between healthy and diseased, but we also need something where there's been no contact with that disease at all. Because even in a population where you've got some individuals that are not doing too well, and some are doing well, chances are those ones that are doing well will eventually become the ones that are going to fall apart, all right? So we don't know if we detect a microbe at those sites, whether it's actually just hasn't quite gotten to that stage yet, all right? So that's why we need those reference sites. We did this very rapidly. It occurred within, I think it was like 4 or 5 days. So basically I sent down materials to do this, and the entire sampling was done within a 24-hour period. It was really, really impressive, and many, many thanks to Alwyn and others here on Saba for facilitating that. And then the samples, because we have the capability of doing molecular biology approaches, things working with DNA and RNA, they sent the samples back to Ithaca, New York via FedEx, which I understand it's really expensive to send stuff back. You should try sending a 70-pound big box back to the U.S. I think it was about $2,800 worth of shipping. All right, so one of the problems with a disease like this is you really have no idea what you're looking at, okay? We need to collect specimens from as wide a suite of samples as we can possibly get so as not to bias the investigation. If we take just spines, we might be missing it if this thing is mainly affecting the internal organs of the animal. And so we came up with this sort of strategy. We took samples of tissues, like the body wall, spines, gonads, intestine, coelomic fluid. That's just the blood on the inside of them. We took samples as well for veterinarians to look at, because what we specialize in is looking at microbiology. Veterinarians have good training in being able to recognize abnormal tissues, which otherwise we don't know what we're doing. Biochemistry as well. We wanted to understand where there's sort of toxins, where there abnormal sort of environmental conditions which might be reflected in the tissues as well. And then we also wanted to see what was in the water column as well around these animals. All right, was it weird plankton that was in the region? Was it, something else, some chemical that came into the environment? So all of these samples were collected at these disease sites and at these reference sites. Very big bunch of investigators who did that. You can see all of them at the bottom there, in a total of 11 different jurisdictions. So getting to the results, what did we find? So the first thing that we do is to see Were things weird at the time? Okay. Were there weird conditions that might have been happening? And so obviously we can't have people in every single place where this disease occurs monitor every single, parameter we're looking at. So oftentimes what we do is we turn to satellite imagery, all right? And using satellites, which are floating up there, you can actually infer what the temperature of the water was, as well as the amount of algae that was in there, all right? So on this little animation, which I'll click go on in just a second in the top left-hand side, what you're looking at is actually the abundance of chlorophyll A. That's a phytoplankton pigment. It's the same thing that gives these leaves their color green. And it tells you about whether there was a lot of algae in the water. And then it will follow on with a temperature inference. So more red colors are the ones that are hotter. And so we look to see whether there was anything that was unusual. So basically, these are the sites that were affected. As they turn red, essentially, that means that the disease first appeared. Unfortunately, there was a lot of cloud cover, as sometimes there is in this region. Satellites can't see through that, so unfortunately we can't infer it. But the point is that there didn't seem to be any unusual high-temperature waters, nor did it appear that there were any unusual blooms, except perhaps in the southern part of the Caribbean, where there was a weird sort of mass of water that came off of the Amazon River plume. The Amazon and the Orinoco rivers put out extraordinarily high amounts of fresh water. They stay on the top of the water because they're relatively buoyant, and then they come into the Caribbean region and they shoal onto islands like this, and you probably see visibility decrease and weird things in the water. Some of the sites that were affected, we had reports of what they were describing as goopy water or gelatinous water. Okay, so this is taken by my colleague Mariah Sevier at the University of Virgin Islands, and she saw a whole bunch of little sort of hair-like structures that occurred at exactly the same time as this disease. We also have algae, things like sargassum, that come on shore, they start to decay, cause the water to turn brown, and lots of material in the water. But there were lots of sites where this disease occurred where none of this was reported at all. All right? So the point is, nothing really consistent that we could say from an environmental standpoint, that's it. All right? So that led us to look at other things, which is, of course, microorganisms. Now, just to give you some idea on the different types of microorganisms that constitute the microbiome, and this includes us, it includes all animals, it includes Bella the parrot behind me as well, believe it or not. It's being very quiet at the moment. So animals in general are inhabited by billions and billions of these things. They're on your skin, they're in your digestive tract, in your lungs, all right? Sometimes even in your blood too, which is really weird. They include things like bacteria, all right? And things also called archaea. They're broadly the same kind, they look the same. They're actually evolutionarily very different, but they make up the vast majority of cells which are in your microbiome. We then have things like microbial eukaryotes. These are things that are tiny little protozoa that are similar to fungi, and there's a couple of images of them there. We also have viruses as well. We generally lump them in with the microbiomes because they're tiny, even though they're not technically alive. There's lots and lots of them associated with even healthy organisms. And so the animal microbiome is what we focused on. We needed to understand what different types within these groups there were. Unfortunately, the approaches that we use to investigate them are very different from bacteria to microbial eukaryotes to viruses. We actually had to go after it with multiple different techniques in order to understand what was different about them. So I'm just going to step back. I have a lot of training in molecular biology as well, but the main sort of take home from this is that we need to look at their DNA or their RNA. We've heard a lot about messenger RNA as a sort of tool for treating COVID or vaccinating against COVID. You can think about it like this. DNA is the core sort of genetic information storage in all living things, okay? It's something which is relatively stable in all cells, but it itself doesn't do a lot. It's inherited from generation to generation, but it's not what actually causes proteins and other cells to be made. That comes down to RNA, and RNA is made from DNA as something called a transcript, and that is through a process of transcription, all right? And then after that, that little transcript is made into proteins, all right? And proteins are the building blocks of cells. This is known as the central dogma, all right? It basically goes from DNA into RNA and to protein. And so when you look at microorganisms, we have to be able to sort of look at these different components in order to understand what the diversity of organisms are actually there. And so down the bottom, I've given some really long technical names for what we did to investigate this. So basically on the DNA side, we looked at all the viruses through viral metagenomics, all right, and amplicon sequencing for the bacteria, and transcriptomics for looking at the host. So these are tools which essentially are taking all of the DNA and all of the RNA out of everything that's there, and then putting it through a machine and trying to understand what was there originally. Okay, it allows us to identify the different types of organisms that are there. So we started off with this thing, which is transcriptomics. So that is a way of saying what organisms are actually doing. Okay, and the way that we do that is we take some normal urchins and some abnormal urchins. We extract this messenger RNA from them, and that's all the signals that the DNA is being made into that tells the basically the host what sort of proteins to make. So it tells you about what they're actually, experiencing in the environment. We put it through some very expensive things called Illumina sequencing. Took a long time to analyze on a computer, and essentially this allows us to say what they were doing. And don't get scared at bar graphs, all right? I'm going to digest it for you. So in this graph, what we have are all of these different categories of genes, which essentially are broken down into functions. And so these are essentially like immune function, replication and repair of the DNA. There's also other sort of biogeochemical related genes, things related to nutrient cycling. And in the green, you can see are the animals that are grossly normal, and in the red are the animals that are grossly abnormal, the ones that are diseased. What this tells us, just broadly looking at it, is that essentially the gene expression profile was that they're growing more slowly, they're responding to something which is invading them, okay? We know that there's this thing called complementation, which can happen when a foreign little thing comes into the animal, and then that they were dying. All right, it wasn't really that informative. I mean, it's not anything that we wouldn't have known by looking at them, but that part about actually responding to something foreign being in them was fairly significant because it gave us our first indication that this was actually maybe a microbe that was involved. We then turned to bacteria, and to do this, we did something called amplicon sequencing. This is where we take DNA out of the healthy and diseased urchins. We extract it. We use the polymerase chain reaction, PCR. You've probably seen it in sci-fi movies or detective dramas as a way of detecting, whether somebody's responsible for a crime. It's almost the same thing. And it allows us basically to separate out and to identify all the different types of bacteria that are there. When we did that, this is another type of bar chart. At the top are the diseased ones. Middle is the ones that are healthy at the same site. And at the bottom are those reference sites. The normal and affected site and the reference site look broadly the same, right? There's sort of the same distribution of colors. Each one of those colors represents a different bacteria. At the top, though, you can see two things that pop out. We have this big green thing at the front. This is a type of bacterium which is normally a type of bacterium that lives within other eukaryotic microorganisms, all right? Big clue there as to what was going on. We sort of didn't realize it at the time. That brown bar is a type of bacterium which normally just eats decaying animals, all right? So that doesn't really tell us much about what was causing it. But this actually turns out, keep an eye on that, that turns out to be very significant down the line. We also looked at viruses, and looking at viruses and their diversity is actually quite challenging. They don't have any sort of thing that we can target with the polymerase chain reaction. We have to basically extract all of their RNA or their DNA, chop it up into tiny little pieces, put it through a really expensive sequencer, reassemble them, so line them all up so that we can identify what different types of viruses there are. And essentially, that's what we did with a few of these specimens. And what we found are really, really boring viruses, okay? I've been doing this for 20 years, and all of these different types of viruses that we detected, they're called disistraviruses. There were some pico-RNA viruses as well. They got funny names like that. Essentially, these are unlikely to be pathogenic. They're viruses, but they're also super, super common in really healthy animals, and so we kind of ruled that out as a possible effect. And then we turn to eukaryotic microorganisms, these fungi-like protozoans. And the way that we did that was to look at expressed genes that we had from healthy and diseased animals, and this was our first clue. When we looked at all of these different types of tiny little eukaryotes, there was a ciliate there which was more represented, or actually only represented in some cases, in the abnormal urchins, and it was absent in the healthy urchins. And that was our aha moment, all right? After spending so long, so much money to do this with DNA sequencing, I ran down to my lab, I went to the microscope and looked down, because these things are really big and they're really obvious when you look them there. And I realized that I wasted about $8,000 worth of sequencing, because I could have just used a microscope to look at them. Okay. But I remember going to the microscope and thinking, oh, that's it. You know, I didn't even need to identify it. It was really, really obvious that these things were taking over the blood. So what is this thing? This is Phylaster apodigitiformis, okay? This is a type of single-celled eukaryotic organism. It's called a ciliate because it's got all these tiny little hairs which are all around the cell. You can see all those little hairs on the left-hand side there. We turned to our veterinarian colleagues who were doing an independent assessment of it. We didn't talk to them during our assessment because we don't want to say, hey, look for this, and then suddenly they find it, and we're totally biasing the investigation. And sure enough, 2 of the veterinary histopathologists found exactly the same thing. This, you see on the right, this is a thin section through one of the spines. And all of these little dots are all this Phylaster apodigitiformis, which are all loaded within the spine. Diadema has hollow spines, okay? Perfect place for this ciliate to exist. And it turns out that's like a hypodermic needle into the animal itself, all right? So really kind of a dramatic thing that we had. So the next question is, was it the same thing across the whole region? Was it exactly the same thing, or were there multiple different ciliates causing this? And to do this, what we did is we analyzed their DNA from many different jurisdictions. This is called a phylogenetic tree, and the idea here is that the things which are located really close to each other horizontally are the ones that are most closely related. Those things which are really far away from them, so for example, this thing at the bottom is really distant from these things at the top, means that you can identify close relatives. And what you can find from this tree is that all of these different jurisdictions here, all right, we had them from Puerto Rico, from the Virgin Islands, from Grenada, all the way up into Florida. They're all exactly the same, okay? So this was it. This was something that we really wanted to focus in on and basically demonstrate that it was actually a pathogen. We also did some more analysis of how many of these ciliates there were in different tissues. And so this is called a box plot. On each one of these panels, in gray, you have the diseased animals. To the left of those, you have the animals that are healthy at the affected sites, and then to the very left side, you've got reference organisms. At the top, we have body wall, which is basically their skin, spine base, so that's that little chunk of material at the bottom of their spine. The blood of the animals, their gonads, and so on. But one thing, you don't have to be a scientist to figure this out here. Those gray bars are really high compared to all of those white bars, okay? This was definitely associated with this condition, all right? So, armed with that, we were very excited because we never see this, all right? This is the most obvious thing I've seen in disease investigation in a really long time. Thank goodness. So what we needed to do then is to be able to experimentally test whether this pathogen can actually generate the disease. And so we waited. We waited for 4 or 5 weeks. We were waiting for it to cross into U.S. territorial waters because transporting cultures, living microorganisms across international boundaries, don't try it, is my advice. We're talking like this much paperwork and 6 months of approvals. So fortunately or unfortunately, it actually showed up in Key Largo about 6 weeks after it started to show up elsewhere. The folks down at the U.S. Fish and Wildlife grabbed a diseased specimen. They did a draw. They took out their salamic fluid. They put it into some culture medium, which if you're interested, the culture medium is really boring. It's actually just seawater and a grain of rice. And then essentially after 2 days, we had our bug, okay? Amazingly, because I have the worst green thumb on the planet, I've tried to cultivate many bacteria, many microbes in the past. This is the first one I actually got. So all the stars were aligning with this whole investigation. So now we had a culture. What could we do with it? Well, you needed to infect some animals to see whether we could generate the same response. And so also by good fortune, the Florida Aquarium, Josh Patterson is a professor there, is involved with aquaculture of diadema. And they had access to a huge number of them, so they gave us 10. No, they gave us, fortunately, they had 10 that we basically treated with this diadema. We actually sprayed this ciliate on their surface, and then we had 10 that we served as controls. And the reason we have controls for this is that they might die from just really poor water quality. I mentioned I've got a terrible green thumb. I'm also terrible with animals in aquariums, so you need to have that control for it. And then we looked and we waited and we saw whether there was any response. And it was actually one of those classic cases of I would go into the lab at 5 o'clock in the morning. I had a key to the lab. This is at the University of South Florida. And I'd report by text message to my colleagues, nothing yet, nothing yet. And then one morning I came in and half of the animals basically had lost their spines and blown themselves apart, which were treated. The healthy animals, totally fine. Okay? So this was evidence that this is definitely responsible for this condition. We even took those animals that were abnormal, that were diseased, we re-isolated this ciliate, and then we basically had what is known as Koch's postulates. So Robert Koch was a scientist in the late 1800s who invented a set of criteria that you basically, a set of steps that you have to go through to definitively prove this thing is a pathogen. All right, so we can definitively say that this was caused by this one thing. So why is this significant? So first off, this is the first time that any ciliate has been found to be responsible for any mass mortality in any organism. They're known for fish, sort of aquaculture, as being kind of pesky, and but they definitely don't cause everything to die off. And so it was like, wow, okay, so this is really interesting. It's a new phenomenon that we're describing. And the other really cool thing that came out of this, as I mentioned, we got this really cool international network of scientists across the region that previously did not exist, all right? Mostly facilitated through this Atlantic and Gulf Rapid Reef Assessment, a few other partners as well. And so we can tackle diseases in the future. There's a lot of outstanding questions that we have about this, all right? This is a little video that I'll show you looking down a microscope. You'll focus in on these cells that are moving around. That bits of tissue that they're actually eating, that's diadema tissue. And one thing that's really interesting about this group of organisms is that under most circumstances, these things are eating bacteria. They're not interested in diadema. But under the right conditions, they seem to target diadema. In fact, they became pathogenic. So we're still trying to get at why this actually happened to begin with. What was it that made them shift their normal diet to going into this pathogenic phase? And so that's something that we're working on right at the moment. That's just another image. Oops, I had to click twice. And this leads us to our second question, which is why now? Has something changed recently? Is this an invasive pathogen? A question that we have very often is, where did this thing come from? I always say the Wuhan Institute of Virology because we just have no idea at all. And that doesn't really matter anyway, all right? But we can ask interesting questions about, can we actually go out and find it in nature? And that's actually what we're doing today with the activity. We were going out and sampling different surfaces, different invertebrates, to see if we can actually detect this pathogen out there now. So in a series of trips that we've done in the past, we've gone out, we sampled everything from boat hulls through to coral surfaces, through to sea fans, I don't know what that is, some algae there as well. We really didn't have any expectation that we were going to find this thing in nature. It seemed to have such a tight association with diadema disease that we really didn't think that we were going to find it out there. And surprisingly, we did, all right? I'm not really that familiar with a lot of the corals in the region going into this, so sending somebody like me out to go and swab animals, I always go for the pretty ones that are really obvious. And in Aruba, which is the first place I went, I just went for the pink ball one, okay, as I called it originally. Turns out that is actually the major host for this pathogen, all right? It's a type of coral called Siderastria siderea. Remember that photo I showed you earlier on? It's the same thing. This is a weedy type of coral that occurs in harbors and ports and also occurs in pretty adverse water conditions that other corals don't like to live in. And so we tested everything. We tested other echinoderms. We tested, mollusks, plants, sponges, abiotic surfaces, that's just saying rocks. It is like orders of magnitude more on those corals, all right? So somehow this ciliate probably normally occurs on this coral, and then through whatever conditions that were present in the beginning of last year, jumped ship and became a pathogen in diadema. But we still don't really know exactly what that sort of trigger was that caused that. Some alarming news happened since this time, since it's affected the region. All right. So first off, it suddenly appeared late last year after it had been in the Caribbean in the Eastern Mediterranean. So the Eastern Mediterranean, you're probably saying diademas over there. Well, in fact, it is an invasive species around Turkey, Greece, also the coast of Israel, and other places in the Levantine. And essentially, all of a sudden, starting in about October, everything started to die. Okay? And then we were watching and waiting because that might have been something totally different. It might have been a different type of microbe. It suddenly turned up in the Red Sea, and then it made its way all the way around to the Gulf of Aqaba, which you can see is the other side of the Sinai Peninsula there. The only way it can move from there to there is via boat traffic. There's no other way that it can go, except maybe a bird or something that picked it up on one side of the Sinai and went to the others. And really, what is very alarming is that just in the last week, it's also showed up on La Réunion Island, which is off of Madagascar, well beyond any sort of current patterns that might have brought it there. This is definitely a ship-transmitted microbe. Currently, what we're trying to do is get information out to major shipping companies to improve their best practices, to try and encourage them to not dump your ballast water within ports, do it sort of well offshore. It's not always easy to do that. There's other things that we're investigating. For example, the firefighting well on big ships, they usually have this big sort of tank that they use for firefighting purposes. Exchange that as well. Try and sort of not have this big incubator on your ship that you're basically moving things from one place to another. And in case you're wondering, yes, it is exactly the same microbe. In the last week, we received word that they have confirmation that Phylaster apodigitiformis here in the Caribbean is genetically identical to the strains that are affecting over there. So, a bit alarming. And just to get back to this coral phenomenon, where it came from in the environment, we actually ran an experiment this summer where we took corals of 4 different species, we put them together into the same tank, and then we basically poured some of this ciliate sort of culture in there to see whether it would gravitate to any particular one. And the answer is it most definitely does. It goes directly to that one species of coral, the Siderastria siderea. All right? So we have lots of evidence that this thing is normally kind of on this coral that's out there. We just don't understand exactly what's causing it to suddenly come off and become pathogenic to diadema. Another question, how long has it been around for? And the answer is we don't have time travel yet. We're still working on that. But essentially we can look back at genetic repertoires of animals that have been sequenced from the region, and we do find it back to at least about 2019. What's really interesting is when we talk about where Phylaster came from, I did a lot of work on something called sea star wasting disease on the US West Coast. It resulted in 98% of the sea stars being wiped out of particular species. Can confirm this ciliate was there back in about 2013 to 2015. And it's just a little too coincident that basically you have this happening immediately after that's kind of undergone this big bloom. So it may have come from the West Coast. It might have also been here as a natural microbe. We just don't know. So just to summarize, we have very rapidly identified the cause of diadema mass mortality across the Caribbean. It's caused by this thing called Phylaster apodigitiformis. Seems to be a very highly prolific, virulent pathogen existing in environmental reservoirs, things like corals, and possibly moving between wide geographic regions and things like, boat traffic. What can we do about it? So this is the next phase of the investigation. We have colleagues at the U.S. Geological Survey who have started to try and treat the actual ciliate with various different veterinary antibiotics and that. And the good news is they actually have 2, which are very sort of common types of treatments for infections by protozoa. The unfortunate thing is we don't have any live diadema to try it on. So while they kill Phylaster, we're not sure whether it will also kill diadema. But that's something that we're hopefully going to get to very soon. And hopefully we can come up with some treatments. And I just want to take a moment just to thank people in my lab. So my graduate student there, Brian, is the person who's done a lot of the work that I just presented. He's from Puerto Rico originally, and he's been thoroughly engaged with just about everything. You've got Brandon as well, my technician. A lot of undergrads who work with me as well. It takes a village to do this kind of work. And then collaborators from across the region, including Marilyn Brandt just up the road at UVI. So, and of course, Alwyn here. So I will say thank you very much. Hope that was useful. Happy to take any questions about this.
So, there's no way to know if this was the cause of the age die-off.
Okay, is this live? So basically specimens were pretty much wiped out. Oh, I'm sorry. The question was, is there any way to know whether this was responsible for the early 1980s die-off? So in the early 1980s, we didn't have access to a lot of the tools that I just gave you some information about. Things like DNA sequencing were either incredibly expensive or they took forever to do, and it was just not practical to apply them. A lot of the specimens that they took were preserved in things that were not really amenable to these new approaches that we have. And then, like I said, some of the specimens were taken, but they were lost in, for example, freezers that would have been very useful to us. Those freezers just go offline when a hurricane comes through, and nobody was able to sort of disperse them anywhere else. The really interesting news is we have come across some specimens just in the last year from about the same time from those places which were affected, from the Florida Keys and from Belize. We have looked for this ciliate, and we can say that yes, it was there back then. Whether it's related to the disease or not, we don't know. And actually, we may never know. It's really, really hard to make an association from just having a microbe there and a disease there. But, and it's unfortunate the ciliate doesn't leave any other sort of scars or anything on the hard tissues that otherwise we could say, oh, look, it's doing the same thing back then. So we probably will never know, unfortunately. Any questions? Yeah? Sure. So the question was about 40% of the specimens that we had in those experiments where we added the ciliate to it did not die, even though they were exposed to it. And does that provide us any clues as to, what's causing that for a start? And secondly, is that something we can harness in the future? So the simple answer is it could be that they have some genetic resistance to it. There could be some immune function that's different about those 40% that we, are not accounting for. We haven't really been focusing on that as yet with our research. That's something that we're hoping to do next. We actually are doing, as we speak here, we're doing that transcriptomic approach to compare those survivors to the ones that have died. And there might possibly be some clues from that might aid in selecting them for breeding, for example, here on Saba. But that stuff really takes a long time, and hopefully we have some answers pretty soon on it. But it's quite possible that they had something inherent about them that led to them being more resistant. In the same way that there's actually populations out there right now of diadema that are totally healthy, and they were somehow resisted this whole thing. Any other questions? Okay. Thank you very much. Thanks for listening. Thank you to the hosts, and thank you to See and Learn.
Thank you, Ian. And now we will be setting up for dinner reservations. So if everyone could grab their chair, please, and take them out and back that way, that would be very helpful. Thank you all for coming.